Researchers in China have achieved the first live birth of a chimeric primate, a monkey with cells from two genetically distinct embryos from the same species. The study, published in the journal Cell, could have implications for genetic engineering, conservation, and other biomedical research. Chimeras are single organisms composed of the cells of more than one genetically distinct individual. They are associated with Greek mythology. Among the best-known examples in mythology are griffins, half lion, half eagle, and the three-headed monster Chimera itself, part lion, part snake, part goat. But chimerism is not just a myth. It exists in nature and through human intervention. The birth of a chimeric monkey with glowing green fingers and eyes may sound like something out of a horror movie, but it represents a major scientific breakthrough. Researchers in China announced the long-sought birth today, sharing images of a long-tailed macaque whose cells are made from an embryo injected with other, genetically distinct cells. Stem cells modified to glow green were injected into a number of marula embryos, those that are 4 to 5 days old, to help identify whether or not the additional cells had multiplied. The contribution of stem cells in different tissue types ranges from 21% to 92%, the Chinese team said. Researchers have long attempted the process in primates, having previously created chimera in rats and mice. In 2012, scientists created 3 rhesus monkeys that were technically chimeras, but the embryos were created at the earlier totipotent stage, where the cells can evolve into anything, including the placenta. The study, published in the journal Cell, used later-stage pluripotent cells, meaning they have the ability to differentiate into all the types of cells needed to create a live animal. This research not only has implications for understanding naive pluripotency in other primates, including humans, but it also has relevant practical implications for genetic engineering and species conservation. Specifically, this work could help us to generate more precise monkey models for studying neurological diseases, as well as for other biomedicine studies. The team first established 9 stem cell lines removed from early-stage embryos, cultivating them to enable an enhanced ability to differentiate. At this stage, they were also labeled with a green fluorescent protein, so the team could easily see which tissues had grown from the added stem cells. One particular subset of stem cells was then selected for injection into a number of 4- and 5-day-old embryos. The embryos were implanted into female macaques, resulting in 12 pregnancies and 6 live births. The analysis confirmed that 1 monkey that was born alive and 1 fetus that was miscarried were substantially chimeric, containing cells that grew out of the stem cells throughout their bodies. Both were male. The Chinese team also confirmed the presence of stem cell-derived cells in the testes and in cells that eventually develop into sperm cells in both animals, meaning the genetically modified cells could be passed on to future generations if the animals were to breed. In 2019, researchers in China created the world's first human monkey chimera, raising significant ethical concerns.